Hello everyone. Today we will talk about different ways of regulation of metabolic reactions. All the metabolic reactions in body, they are well regulated and well controlled, and that's a physiological requirement also. If for example, if you do not control synthesis of glucose in gluconeogenesis, so all the precursors will be converted into glucose, so that no intermediates are available for further reactions to be carried out in the cell. So that's why each and every reaction, they are very well regulated and very well controlled in a cell. And today in this lecture we will talk about some of the very common methods which are used by cell in order to regulate metabolic reactions. So there are at least 6 ways of regulation of metabolic reactions, how those enzymes involved in metabolic reactions are regulated. So first one is product inhibition. So product inhibition means product of a reaction is inhibiting same enzyme. So when product is in high concentration, for example, you have a substrate, this is being converted into some product and is done by catalyzed by enzyme E. So when concentration of product is high in the cell, this product binds to this enzyme and inhibits its activity. That means product is already available in sufficient amount in the cell, so no further conversion of a substrate to product is required. So that's one way to regulate enzymatic reactions and metabolic reactions. Another method is mass action ratio. In mass action ratio, that's applicable basically on reversible reactions. So for example, if A is being converted into product, so the concentration of A and P will decide whether the reaction should proceed in forward direction or backward direction. Another method is allosteric regulation. This is also a very common method which regulate enzyme activities in different metabolic reactions. In this mechanism, and those enzymes which are regulated by this method, they have a separate site besides substrate binding site on their surface, and some ligand molecule, some chemical, when it binds to that additional pocket or additional site on the enzyme surface, that change activity of that enzyme. So that we'll discuss in detail in later on. Then there are regulatory proteins. As per the requirement by cell, some proteins are synthesized which work inhibitor for particular enzymes, and when these proteins are present in cell, those proteins bind to the enzyme and make that inactivated. Another method is covalent modification. The best example of covalent modification is phosphorylation. When a phosphate group is transferred from a nucleotide phosphate to an enzyme, that enzyme becomes activated or deactivated by that action of kinases. You call them kinase enzyme, which transfer phosphate group from a nucleotide triphosphate to a target enzyme which is involved in a metabolic reaction. Then the 6th method is gene expression or transcription level control. For example, if a gene is not required or a product is not required by cell, a substrate is being converted into product by enzyme E, and if P is not required, it means cell can stop this production of this enzyme also. So enzyme production also can be regulated. And how enzymes are being produced? Enzymes are proteins. So they are actually produced from DNA. So they have a gene, a segment of DNA which encode for that enzyme, and that gene is expressed into messenger RNA, and that messenger RNA is converted into protein by translation process. So in this method, the gene production is controlled. How the protein is being produced, how is protein or enzyme is being produced from by expression of that DNA segment, what you call gene, that is regulated. So we will talk about these methods in detail now with examples like product inhibition. So this is an example of hexose monophosphate pathway or pentose phosphate pathway, where glucose is converted into pentose sugars, pentose phosphate, and there is a regulatory enzyme here which convert glucose 6-phosphate to 6-phosphogluconolactone. That enzyme is called glucose 6-phosphate dehydrogenase, and that's the regulatory enzyme of pentose phosphate pathway. As a result of this action by glucose 6-phosphate dehydrogenase, NADPH is produced, and this NADPH works as an inhibitor to the same enzyme. So when NADPH is in high concentration in cell, it means it's no longer required in further concentration. So the production of NADPH should stop. So that signal is taken up by NADPH, and high concentration of NADPH bind to the same enzyme and inhibits the activity. So here NADPH work as a competitive inhibition. So in this case, the enzyme called glucose 6-phosphate dehydrogenase, it has two sites. For example, you have something like this, the one site where glucose 6-phosphate bind, and another site is for NADP+. NADP+ is a cofactor of this enzyme. As a result of this reaction, NADP+ is converted to NADPH, and this NADPH work as a competitive inhibitor for this enzyme. It means when NADPH is in high concentration, it binds, it replaces NADP+, and it does not allow NADP+ to bind to this enzyme. As a result, this reaction cannot proceed. So that's how NADPH, when present in high concentration, it works as a competitive inhibitor, which is a product of this same reaction. So that's an example of product inhibition. So that occurs in many cases in metabolic reaction. There are many enzymes which get inhibited by their own product when product is there in high concentration. Another mechanism of regulation of metabolic reaction is mass action ratio. So the example here is again from oxidative reactions of pentose phosphate pathway, where a pentose sugar ribose 5-phosphate and another pentose xylulose 5-phosphate, they react together and form several variable carbon number sugars, and eventually you have glycerol 3-phosphate and fructose 6-phosphate. So these are product of these reactions. These are all reversible reactions. So here the concentration of these products or the substrates, those determine whether reaction will proceed in forward direction or the backward direction. For example, if cell has ample amount of glucose 6-phosphate and glyceraldehyde 3-phosphate, in that case these reaction will be reversed and the product will be ribose 5-phosphate and xylulose 5-phosphate. Similarly, if cell has sufficient concentration of pentose sugar in form of ribose 5-phosphate or xylulose 5-phosphate, the reaction will proceed in forward direction and you have synthesis, more production of fructose 6-phosphate and glyceraldehyde 3-phosphate. Another mechanism of regulation of enzyme activity in metabolic reactions is by allosteric regulation. The enzymes which get regulated by allosteric mechanism, they have two sites like here. So one site is the active site where substrate binds, and another site which is different than the active site or substrate binding site is called allosteric site. So any allosteric molecule bind to this site, and there are two types of allosteric regulation. One is called allosteric activator, another one allosteric inhibitor. So when any allosteric activator binds to this site, it changes conformation of the enzyme in such a way that enzyme can now efficiently bind to the substrate and can catalyze conversion of substrate into product. But in case of allosteric inhibitor, when an inhibitor molecule bind to the allosteric site, what you call allosteric inhibitor, in that case conformation of enzyme is changed in a manner that the substrate can no longer bind to the active site. Hence the enzyme activity is controlled, is inhibited. So some example of this regulation is mechanism of regulating phosphofructokinase 1, which is one of the key enzymes of glycolysis, and same time another enzyme which is used in to reverse the same reaction of glycolysis in case of gluconeogenesis, where glucose synthesis takes place by reversal of reactions of glycolysis. That enzyme is FBPase, fructose bisphosphatase. And these PFK1 of glycolysis and FBPase1 of gluconeogenesis, they both are regulated by same compound, what is called fructose 2,6-bisphosphate. That we have discussed in earlier lecture on glycolysis. So fructose 2,6-bisphosphate bind to both the enzymes, but it has different roles. In case of PFK1, phosphofructokinase, it increases activity of PFK1, that we can see by the red line. So when fructose 2,6-bisphosphate is present, it enhances activity of PFK1, so it works as an allosteric activator for PFK1. At the same time, when same molecule binds to FBPase1, which is an enzyme of gluconeogenesis, it inhibits activity of FBPase1. So the same compound is activating glycolysis and same time it's inhibiting gluconeogenesis by inhibiting activity of FBPase1 and activating PFK1. Another example of regulation of metabolic reactions is by production of regulatory proteins. In this case, some protein inhibitors of enzymes, they are produced by the cell with response to some external or internal stimuli depending on the requirement by the cell, and those molecules, they form complex with the target enzyme and inactivate it. So for example, in case of glycogen metabolism, that's what we have seen in the earlier lecture on glycogen metabolism, where epinephrine in muscle, it activates pKa1, and this pKa1 in turn activate inhibitor of phosphatase, which is called inhibitor 1 here, by transferring phosphate group on it. So this inhibitory protein get activated, and when PP1, which is a phosphoprotein phosphatase that is present in cell, this inhibitory protein form complex with phosphatase. As a result, phosphatase cannot work. So that's what we have seen in case of glycogen metabolism, that when PP1 is inactivated, that's the final result of action of epinephrine or adrenaline in muscles, which leads to phosphorylation, activation of phosphorylase, that is involved in catabolism of glycogen. Similarly, in case of glycolysis, there was another example of similar kind where regulatory protein binds to hexokinase 4. In case of liver cell, hexokinase 4 is a glucokinase. So one method of regulation of glucokinase is by production of regulatory protein. So regulatory protein is activated by concentration of fructose 6-phosphate, and this regulatory protein, when it's activated, it binds to hexokinase, form a complex and transport hexokinase 4 to nucleus. And because glycolysis is occurring inside cytosol, so when hexokinase 4 is not available in the cytosol, is being transported to nucleus, so that glycolysis is inhibited. and this is reversed by presence of glucose. When glucose is present, it deactivate, dissociate this complex of hexokinase 4 and regulatory protein, so that hexokinase is transported back to cytosol in order to have continued glycolysis process in the cytosol. Fifth mechanism of regulation is by covalent modification, and phosphorylation is a most common method which is used by cell in order to activate or deactivate enzymes of different metabolic pathways. And there are two enzymes which are involved in this modification. The one enzyme transfer phosphate group from a nucleotide triphosphate to the target enzyme. You call them kinases. For example, here you have GSK3 in case of glycogen metabolism, glycogen synthase. The activity of glycogen synthase is regulated by GSK3, glycogen synthase kinase 3, which transfer phosphate group on glycogen synthase and make it inactive. And there is another enzyme which is called phosphoprotein phosphatase. Phosphoprotein phosphatase does reverse of the action of kinase. Phosphatase remove phosphate group from the protein. So there are two types of things which can happen in this kind of mechanism. A protein may get activated by transfer of phosphate group on its. Phosphate group is transferred on hydroxyl group containing amino acids in protein like threonine and threonine, tyrosine or serine, and that's why these kinases which transfer phosphate group on these OH side chains, they are of three types. You have serine kinase or tyrosine kinase or threonine kinases. So kinases transfer phosphate group from nucleotide triphosphate to the target protein, and phosphatases remove that phosphate group. So two possibilities are there. Some protein may get activated by the action of kinases. So when you have phosphorylated form of those proteins, they are active. Other proteins, they may get deactivated by transferring phosphate group on their surface. So for example, in case of glycogen synthase is active when there is no phosphate group. So when you transfer phosphate group on glycogen synthase, it's become deactivated. Similarly, if you talk about phosphorylase, glycogen phosphorylase, which is involved in catabolism of glycogen, in that case, the phosphorylated form of phosphorylase is activated, and when phosphate group is removed, the phosphorylase is become deactivated. So it depends on the enzyme whether the activity is being enhanced by presence of phosphate group or by removal of the phosphate group on its surface. Another example of regulation of enzymatic activity in metabolic reaction is by controlling gene expression. We know that enzymes are proteins, and those proteins are synthesized from DNA, the fragment of DNA what you call gene, that is converted into, if you look at the central dogma, the gene on DNA is converted into RNA, messenger RNA to be more precise in this case. That's a process what is called transcription. And this messenger RNA here, it is converted into protein, that is enzyme, that has enzymatic reactions by a process called translation. So when cell doesn't require a product, so in that case cell can suppress expression of this enzyme. And how can cell do that? By controlling transcription. That's why this mechanism is also known as transcriptional control or transcriptional level regulation of enzyme activity. So in that case, transcription is controlled. So when transcription is controlled means there won't be sufficient messenger RNA in the cell, and when messenger RNA is not sufficient, it means the product of messenger RNA, which is protein, that also will present in low concentration. Or if cell require high concentration of enzyme or the product which is result of that enzymatic action, in that case the overexpression of that gene coding for this enzyme occurs, so that the concentration of gene product protein is high in the cell, and when enzyme present in high concentration, naturally it will produce more product which is required by the cell. So one example of this kind of regulation is effect of insulin on gluconeogenesis. When glucose present in high concentration in blood, insulin is secreted. When glucose is already high, it means synthesis of glucose inside the cell from non-sugar molecules is not required because glucose is already there in good concentration. So in that way, insulin inhibit inside cell synthesis of glucose, what you call gluconeogenesis. So how does insulin do that? Insulin will bind to its receptor on cell surface, and that will start a cascade of reaction, eventually activating a kinase called protein kinase B, PKB, and PKB phosphorylate, because it's a kinase, so it phosphorylate FOXO1. FOXO1 is a transcription factor. Transcription factor means which enhance expression of certain genes, and those genes here are PEP carboxykinase and glucose 6-phosphatase. These two enzymes, they are key enzymes of gluconeogenesis. So when FOXO1 is present in cell in non-phosphorylated form, FOXO1 will enter to nucleus where transcription is occurring. It will bind to the target gene site, genes which codes for PEP carboxykinase and fructose 6-phosphatase. And after binding to the region which control expression of these two genes, it enhances expression of these two proteins. So it means FOXO1 is required to have high concentration of PEP carboxykinase and glucose 6-phosphatase. And these two enzymes, they are key enzymes of gluconeogenesis. So in turn, FOXO1 enhance gluconeogenesis, that is synthesis of glucose from non-sugar molecules in the cell. But insulin suppress it. So how does insulin do that? Insulin activate protein kinase B, PKB, and this PKB phosphorylate FOXO1, and the phosphorylated form of FOXO1 is degraded by the cell with the help of proteasome. Proteasome are complex of proteases which remove proteins which are no more required by the cell. So phosphorylated form of FOXO1 is degraded, and when FOXO1 is degraded, it means FOXO1 cannot enter to nucleus, it cannot enhance expression of PEP carboxykinase and glucose 6-phosphatase. So that's what insulin do. So insulin basically remove FOXO1 from the cytosol so that FOXO1 cannot increase concentration of these enzymes PEP carboxykinase and glucose 6-phosphatase, so that glucose synthesis by gluconeogenesis is inhibited. So these are 6 common methods which are basically used by cell in order to regulate metabolic reactions.